Dear student friends, today we shall discuss about the uh, informations regarding uh, proteins and its structures. Okay, so the proteins are the polymers of alpha amino acids. As we know, therefore they are polyamides in nature. Okay, in proteins, different alpha amino acids are linked to one another by peptide linkages. Okay, chemically, the elements carbon, hydrogen, oxygen, nitrogen are present in all the proteins. Some of these may contain elements like sulfur, phosphorus, iodine, and traces of metal like iron, zinc, and manganese, etc. Okay, actually, the proteins are peptides with a very high molecular weights, more than 10,000 daltons. Okay, these are formed by the combinations of more than 100 alpha amino acids. Okay, so normally these amino acids are having two functionalities: amine group and carboxylic groups. Okay, COOH group and NH2 group. So NH2 of one amino acids are linked with COOH of another amino acid. So which will make an uh, amide bonds, CONH bonds. Okay, among these two. So that is why it is polyamide in nature. I mean, 100 more than 100 molecules of amino acids are linked. Okay, they are said to be. These proteins are also. Uh, I mean, considering under. I mean, this is a biomolecules, as we know, isn't it? So the name protein is derived from Greek words. Remember, proteus, which means first. Okay, the name is well chosen because the proteins are ranked first amongst the natural polymers essential for the growth and the maintenance of life. And the proteins make up a large part of animal bodies and are the complex nitrogenous polymeric compounds present in all the living cells. And these are the principal, I mean, components of skin, muscles, and blood enzymes. Remember, also many hormones. Proteins are involved in all the critical functions of living systems, and they serve as nutrients, regulate the metabolism, assist the absorptions of oxygen, and play important role in the functioning of nervous system. Remember, they are provide the mechanical base for the muscle contractions also. Represent the major support material for the body and also helps in the transfer of genetic information. Okay, with this brief information, I can go through. The classifications of these proteins. I will go through the classifications. Okay, so as we know, there are proteins are classified in various ways. I hope as per as your syllabus, I mean KUD syllabus, I mean 6M BSc 6M KUD syllabus have included, the primary structure and secondary structure of proteins. Okay, so it is a part of, the structures. Okay, there are, as I told, proteins are classified in many ways. So in the broader sense, I hope these classifications of proteins on the basis of molecular structure, there will be two types. One is fibrous proteins and globular proteins. So again, the classifications of proteins on the basis of hydrolysis product or the compositions. Remember, simple proteins, conjugated proteins, and derived proteins. So there are many ways of classifications are there. I mean, I will restrict only about the proteins. I mean, classification of proteins based of molecular structure. In this molecular structure, basis of molecular structure, only two types of structures are there. One is fibrous proteins, another one is globular proteins. Okay. So fibrous protein means they are the thread-like, molecules. Remember, so we tend to lie the side by side to form fibers. Okay, so the molecules are held together by hydrogen bonds. Okay, they are generally insoluble in water. And globular proteins concerned, whereas the globular proteins are soluble in water. Remember, these are folded into compact units that often approach the spheroidal shapes. Okay, so here in which the intermolecular forces, that means intermolecular forces means between the molecule. Remember, in them are weak due to the smaller area of contact between the molecules. These are soluble in water. Examples albumin and plant proteins, etc. As far as your fibrous protein examples are keratin and collagen. Kera- Keratins are present in hair, skins, nails, and feather, etc. Collagens present in bones. Okay, these are fibrous. Globular proteins are albumins. Okay, so and plant proteins, etc. So, as far as these functions of globular protein concerned, so the globular proteins catalyze all the biological reactions. That means they are involved in biochemical reactions. They catalyze the biochemical reactions. Remember, and they regulate metabolic process. For example, insulin hormones maintain blood sugar level and act as a antibodies also, which protect the body from the effect of invading species. Remember, and transport of oxygen from lungs to various tissues through bloodstream. Example, hemoglobin. Remember, these are the functions of globular proteins. Okay, so with these brief information, one could, I hope, distinguish these two proteins very clearly. Okay. Okay, as we discussed in the earlier slide that proteins are existed in two types as far as its classification says. Okay, based on its molecular structure or compositions, one could classify the proteins into two categories, which are fibrous proteins and globular proteins. So these two are the different, um, protein structure which we have distinguished, isn't it? So, in the following ways. So, fibrous protein in the sense they are the long chain of polypeptides that occur in bundles. Okay. So, globular protein concerned, whereas globular proteins roughly spherical in shape. Okay. And second distinctions regarding the protein structures is The fibrous proteins are insoluble in water, whereas the globular proteins are soluble in water. Okay, so the examples for fibrous proteins are keratin and collagen. Okay, so globular proteins are essentially all enzymes. Okay, are the globular proteins. So enzymes are acts as an biocatalyst in biochemical reactions. As we know, most of the these biochemical reactions are operated by the bioenzymes only. Okay. So again, the stability concerned, fibrous proteins are stabilized by hydrogen bondings. Okay, so they are linked by hydrogen bondings. Okay, and again, the globular proteins concerned. Here also, these proteins are stabilized by hydrogen bondings only. So intermolecular hydrogen bondings are existed. Okay. So this is what distinguish of fibrous protein with globular proteins. Okay. Okay, now let us move on to the structures of proteins. If we look into the structure of proteins, which are described by the several levels. Okay, so normally which are existed in primary structure, secondary structure, and tertiary structure, and quaternary structures. Okay, primary structure describes sequence and the number of amino acids in the chain, as well as the locations of all disulfide bonds or bridges in the molecule. Then second one is the secondary structure, which describes the regular conformations of segments of protein backbones. In other words, this secondary structure describes how the local regions of backbones folds. Okay, so this is what secondary structures. And third one is the tertiary structure. Tertiary structure means it is a three-dimensional network of entire polypeptides. Okay, so the resulting three-dimensional shape formed by the precise folding and bending of polypeptide chain in the helical coil is referred to as the tertiary structure of proteins. Remember, here the more folding of proteins, the secondary structure and more folding of secondary structure takes place, that is what tertiary structure. Okay. So in single structure itself, one could observe many interactions, covalent interactions as well as non-covalent bondings. So non-covalent bondings like hydrophobic interactions, weak electrostatic force of attractions, and hydrogen bondings, and again disulfide bonds. All are exist in a single molecule, so therefore it will be um, more stable comparing with the secondary one, because here more folding is there. So that is why it will be a three-dimensional network. Okay. And the last one is quaternary structure. So quaternary means that the protein has more than one polypeptide chain. Obviously, it has a quaternary structure. This quaternary structure of protein is the way the individual protein chains are arranged with respect to each other. Okay. So these quaternary structures are stabilized by ionic bond, hydrogen bonds, and hydrophobic interactions. Okay. So, for example, hemoglobin is such a protein with the four polypeptide chains, and these chains are linked together by covalent bonds. Remember, in which four subunits are available, which are linked together by the covalent bonds. Okay, so as like it is also called as a tetramer. Four subunits. I mean four. I mean subunits of amino acids are linked together to give. I mean hemoglobin molecule. Now let us move on to the primary structure first. Okay, primary structure of proteins describes the sequence of amino acids in the proteins. Okay, so hence the primary structure involves peptide bonds. Okay, breakage of these peptide bonds or the formation of new peptide bonds leads to the changes in the primary structure of the proteins. It also describes the sequence and the number of amino acids in the chain and the locations of all the disulfide bonds. Remember, on complete hydrolysis of proteins, one could get the number of amino acids which are available, alpha amino acids. One could calculate or one could determine. Okay, and to know the location of all disulfide bonds, one could reduce the disulfide bonds. Okay, so reduction of disulfide bonds gives the locations. Okay, complete hydrolysis gives the number of amino acids. How many amino acids are linked in this proteins? Okay, and the sequence of amino acids concerned, one could get the idea through this sequence of amino acids could be done through terminal residue analysis or the partial hydrolysis. These two methods have been distinguished. Okay, established. With the help of these two methods, one could able to get the sequence also. So overall, I can say that this primary structure describes the sequence, number of amino acids, and the locations of disulfides. Okay, bridges. Also, it includes bond angles, bond length of carbon-carbon bonds in the peptides. Okay, so these are the things which are related to the primary structure. Okay, now the second one is the secondary structure. As I told in the earlier slides, this describes regular conformations of segments of proteins backbones. Remember, so the usually primary structure of protein gives only the nature of linkages of alpha amino acids in the protein chain. However, it also gives no, it gives us no idea about the shape or conformations of the molecule. Remember. So, on the other hand, the secondary structure of protein is related to the arrangement of polypeptide chains that may be a linear or they may get coiled to form a helical structure. Okay, so this is what the secondary structure. Okay, the folding of peptide chains arises due to the hydrogen bonding between the CO and NH groups. Okay, so and as I told in the beginning, so the proteins are undergoing foldings. So to minimize the energy, So the polypeptide chains tend to fold in the repeating geometric structures such as an alpha helix or beta plated sheets. Remember, so they exist in two forms, helical structures and beta plated structures. So the requirement of secondary structures, as far as these two, I mean alpha helix and beta plated sheets, depends on three, aspects. The first one is regional planarity, amide bond resonance about each peptide bonds, okay, which limits the possible conformations of peptide chains. Because this is very important, I hope, because the hydrogen bonding takes place between these two, um, functionality. CO group and C double bond O and NH group of amino acids. So that therefore the regional planarity plays major role in that. Okay, about each peptide bonds, which limits the possible conformations of peptide chains. The second thing, hydrogen bonding between the carbonyl oxygen of one amino acid residue and the amide hydrogen of another one. Okay. So the third aspect will be the adequate separation between the nearby R groups to avoid steric hindrance and the repulsions of like charges. I hope these three are the requirements for the secondary structures. Okay. So I hope as far as this secondary structure concerned, there will be two types. One is alpha helix structure and another one is beta plated structure. Now let us discuss about one by one. What is alpha helix and what is beta plated sheets? So alpha helix. Alpha helix means the backbone of the polypeptide coils around the long axis of protein molecules called alpha helix. Okay. So these helix is stabilized by hydrogen bonding. Remember, so the NH hydrogen of peptide, amide hydrogen it is, remember, okay, of peptide attached to the carbonyl oxygen CO of an amino acid 4 residue away. All the CO points in one direction, okay, and all NH bonds points in opposite direction. So therefore, peptide NH hydrogen of peptide attached to the carbonyl oxygen CO of an amino acid 4 residue away. Okay, because the carbonyl carbon points in one directions and all NH bonds points in opposite directions. So therefore, the E NH of peptide attached to the carbonyl oxygen of an amino acid 4 residue away. Okay, so then the substituents are on alpha carbons, remember, of the amino acids projects outward from the helix, thereby minimizing the steric hindrance. I hope in the earlier slide I told requirements of alpha helix. Remember, so this is very important because uh, if there R groups are very nearer in the structure, they repel each other because groups always repel each other. Remember, so therefore substituent R on the alpha carbon of amino acid projects outward from the helix, thereby minimizing steric hindrance. Remember, and then the alpha helix is the right-handed helix because why it is so, it rotates in clockwise directions as it spirals down because amino acids have L configurations. So most of the, I mean, the proteins are made up with alpha amino acids. They are in L configurations. Remember, okay? And each turn of helix contains, remember, 3.6 amino acid residue, and repeat distance of helix will be 5.4 angstrom here. It is. This is about alpha helix structure. Okay, and not all amino acids are able to fit into the alpha helix. As I told in the beginning, I mean there are three requirements. Otherwise, I mean proteins are not existed in alpha helix. So, for example, proline residue. Remember, for the amino acid whose nitrogen atom forms part of five-membered ring is more rigid than other amino acid, hence cannot rotate to enable it to fit readily into the helix. So that was the reason. So, and the similarly, two adjacent amino acids that have more than one substituents on the valine, isoleucine, threonine cannot fit into the, I mean, helix because steric crowding between the R groups are there. Remember, there must be a less steric hindrance. Then only I think one could expect alpha helix structure from the proteins. Okay. So finally, two adjacent amino acids with like charged substituents cannot fit into the helix because electrostatic repulsion between the R groups will be there. So they must be far away from each other. Remember, otherwise they will be in different side. They exist in different side. Remember. So the and then percentage of amino acid residue coiled into alpha helix varies from protein to protein. But on average, about 25% of residue in globular proteins are in, I hope, alpha helix structure. Okay. So and myosin and alpha keratin in hair are composed almost alpha helices. Remember, this is all about alpha helix. Then go for now. Let us discuss about beta pleated sheet. Beta pleated sheets forms when two or more peptide chains called strands line up side by side. Okay, here one could observe, I mean, two types of structure. One is parallel beta pleated sheet, and another one is interparallel beta pleated sheets. So, in the parallel and beta pleated sheet, two strands are run parallelly. Remember, N terminal parallelly run with, N terminal of one peptide chain parallelly run with the N terminal of another peptide chains. Therefore, they are parallel beta pleated sheets. And another one is anti-parallel. So, N terminal of the peptide chain, one peptide chain is run with, parallelly run with, I hope, C terminal of the another peptide chain. So, therefore, it is called anti-parallel beta pleated sheets. In both these parallel and anti-parallel beta plated sheet, one could observe hydrogen bonding between carbonyl oxygen and NH group of amide linkage of the another sequence. Remember, in both the cases, so hydrogen bonding is the stability factor here. Remember. So the beta pleated sheets concerned. the following characteristics are there. I mean, as I told in the earlier secondary structure only. So there are certain requirements. So these requirements are called as characteristics only. So the carbonyl carbon and NH bonds lie in the plane of sheets. Okay. So then the hydrogen bonding often occurs between CO and NH group nearby amino acid residues. Okay. And then R groups are oriented above and below the plane of sheets and alternate from the one side to the other along a given strand. So this will helps the I mean formations of beta pleated sheets because steric less steric hindrance are very much required for the formations of protein structures. Remember, the adjacent hydrogen bonded peptide chains can run in the same directions or opposite directions. One is parallel, another one is anti-parallel. So parallel means adjacent chain run with the same direction, whereas with beta pleated sheet, adjacent chain run in opposite directions. Okay, so that I have illustrated clearly in the earlier slide. Okay, then go for the tertiary structure of proteins. As I told in the earlier slides, tertiary structure of proteins is the three-dimensional arrangement of all the atoms in the proteins. Okay, so which results from twisting, bending, and folding of the protein helix. Further folding of the secondary structure it is. Tertiary structure means further folding of the secondary structure. I mean, here one could expect, I mean, more folding from this proteins. Remember, so the protein folds spontaneously in solutions in order to maximize their stability. I hope to get the stability only, I hope uh, foldings takes place. Okay, so here the stability factors are covalent bonds, hydrogen bonds, electrostatic attractions, and van der Waals force of interaction. Remember, so hydrophobic. Hydrophobic interaction in the sense the non-polar group forms cluster in the interior of protein due to their hydrophobic nature. Hydrophilic groups, polar groups will tend to lie on the surface of protein molecule. So I hope this is about the tertiary structures. Okay. Okay, this is an schematic representation of tertiary structure of proteins. Here one could visualize the proteins foldings. Remember, proteins fold spontaneously in order to maximize their stability. Remember, here in single, I mean structure itself, one could observe, I hope, all the types of bondings, covalent and non-covalent interactions. I hope hydrogen bonding, disulfide bonds, electrostatic attractions, and hydrophobic interactions in a single structure itself. Remember, so that is why I hope these tertiary structures are more folded than the, I mean secondary structure. And the last one is quaternary structure. So if protein has more than one polypeptide chain, it has a quaternary structure. Remember, obviously it has. Okay, this quaternary structure of protein is the way the individual protein chains are arranged with respect to each So this represents or refers to a special arrangement with respect to each other, okay, of these polypeptide chains of a complex protein. So normally proteins that have more than one peptide chains are called oligomers. Individual chains are called subunits. Okay, based on the number of subunits, they are classified into monomers, trimers, dimers, tetramers, etc. Here it is best illustrated with hemoglobin. Remember, hemoglobin is tetramer. So, four, subunits are linked here. So, they are, labeled in different colors green, orange, purple, blue. Okay, these are all, I hope, subunits of hemoglobins. Remember, so therefore, it will be a tetramer. Tetramer in the sense four amino acid units it is. Okay, four amino acid chain. Okay, they are said to be as subunits. Individual protein chains hold together in three-dimensional conformations by hydrophobic interactions, hydrogen bondings, and electrostatic attractions. And the possible quaternary structures for hexamers are these. Okay, linear, spherical, and egg-shaped structure. Okay, I hope these three are the possible structures of proteins. Now let us discuss about protein denaturations. What is protein denaturations? Protein denaturation means destroying the highly organized tertiary structure of proteins. Is called denaturations. It is what structural changes. Okay, I hope disruptions of non-covalent interactions which are available in the proteins is called denaturations. There are several factors influencing on these denaturations. As we know, if body temperature increases from 37 degree, denaturations starts. Okay, so best example for this denaturations is uh, egg white part of an egg. Okay, so if we heat this white part of an egg, um, or by wrapping, one could, I think, observe denaturation. Remember, so it is best example one can quote. So there are several factors apart from these. By changing pH. Okay, so this will changes the charge which are available on the side chains. Okay, this means this disrupts the electrostatic attractions and hydrogen bondings. Okay, so certain reagents also influencing the denaturations. Suppose, for example, urea and guanidine hydrochloride denatures the proteins by forming hydrogen bond to the protein groups that are stronger than hydrogen bond formed between the groups, because this will these uh, guanidine hydrochloride and ureas are replacing the hydrogen bond already existed in the normal proteins. So that means these are the stronger binding sides rather than the uh, I mean um, hy- hydrogen bonds which are existed early. Remember, so and detergents also, like sodium dadosyl sulfide denatures proteins by associating with the non-polar group of protein. So this these Dadosyl sulfates, I hope, replacing the, compete with the non-polar group of proteins. So thus interfering with the normal hydrophobic interactions which are available. So thereby also one could I think denature the proteins. And again organic solvents or by the applications of heat or agitations. So heat or agitation in the sense which will increase the molecular motions, which can interrupt the attractive forces which are available in the molecule. Okay, so these are the several factors which are influencing on the denaturations of proteins, which are pH, certain reagents, detergents, organic solvents, and heat or agitations. Remember, thank you.